right, today we're on 6A. We're looking at DNA, proteins in the cell cycle, and we'll finish some of that cell cycle, I believe, on next week on 6B. Okay, so here we go. DNA, genes, and chromosomes. Genetics is the study of how characteristics are passed to offspring, and it's a complex study involving several factors that lend toward this. The first one will be genetic factors. What does your DNA look like? Are you tall? Are you short? Do you have curly hair? Do you have blue eyes? Do you have brown eyes? Whatever it is, those are genetic factors. Two, environmental factors. Where did you grow up? What were the conditions? How was your nutrition? Things like that. And then the third one would be spiritual factors. Did you grow up in a house where you believed in God and had regular church attendance, things like this? In other words, those steady things in your life that would keep you on an even keel and help to influence the way you turned out. Okay, so on some of these, we probably may want to put a little more emphasis on genetic factors because, in other words, however, whatever the code for us is makes us who we are, yes? But then again, our spiritual identity makes us who we are too. And then your familia, your family, how you belong in your family, this kind of thing, helps in defining your identity, yes? And so all of these things we're going to see. The biological side of it, since we're looking at biology, we're probably going to spend most time here on that. So what is DNA and what does it look like? What does DNA stand for? We had it right back here? No? We didn't have it. It's a long word. It's a big long protein. It stands for deoxyribonucleic acid. Deoxyribonucleic acid. And it's actually describing, and it's acidic. It's got ribose in it. It's got a nucleic acid in it. Okay, it's just really describing it. What does it look like? The answer to that is we're not exactly sure. We think it looks something like this picture on the left side right here, what is known as a double helix.
There's actually some evidence that proves it might be a triple helix.
Right, that there's one on the outside of it. Yeah. So that's new data, new visual. Since it's so small, it's hard to see it, but we can bounce like X-rays off of it and get an idea like a shadow. And so now we're thinking that maybe there actually is another rung out here on the outside of it. So making it either, if you call it a triple helix at that point, I don't know. Double, I don't know. We'll have to wait and see, okay? So bottom line, what does it look like? We're not exactly sure, but we'll go with our double helix. In 1953, two different scientists were working on isolating DNA from living cells. One was in England, one was in the US. Mr. Watson and Mr. Crick. I believe Watson is the American. Francis Crick is the Brit, I think, if I remember that right. And they both independently came up with they were using similar techniques, and they independently came up with the structure. It's a unique form called the double helix. So go ahead and write double helix somewhere in your paper, please. Okay, if I ask you what does DNA look like, what does it shape like, you'll tell me double helix. It's like taking a spiral staircase, or taking a ladder and twisting it like a spiral staircase. Okay, we're going to make a model of it here in just a little bit, and that'll help you to remember what it looks like. Okay, and we call that a double helix. It contains information in a code form that's responsible for building all of the proteins in a living organism, everything it needs, and transmitting hereditary traits. Okay, so we know that your DNA, you got some from your mom and some from your dad, half and half, yeah? And that's going to give you kind of a mixture of them, and you're going to have certain traits that they may have. If you're tall, you might be tall, if they're tall. If they have certain, physical traits like the color of their skin, the color of their hair, the color of their eyes, they're going to pass that on to you. And we're going to see sometimes there are some traits that are considered dominant, and some that are not dominant. They're called recessive, and therefore, yeah, that's one of them. The hitchhiker's thumb, if you do your hitchhiker's thumb like this and do Giga-Maggie's and it goes all the way back, that is dominant. Some people can't go any farther than just doing that. We're going to look next week specifically. I'm going to have you taste something, and some people can taste this piece of paper and other people cannot. We'll do that next week. And also you're going to do a little review of your family and see a look at everybody's earlobes. Okay, our earlobes are detached or attached. Mine are detached, which makes for better hanging of earrings right there. But some of them are attached to the face, and you're going to check your family out and see what they look like. Okay, I have 5 kids. I have a detached earlobe right here. My husband has attached earlobes. I have 5 children. All 5 have detached. Which one do you think is dominant? Detached.
Say the attached.
Yeah, all of them. The detached. All of them have detached. Okay, but in addition to our DNA coding for those things, it's working in your body right now. All right, what it's doing is it's taking, hopefully you had some lunch. Do y'all not eat lunch? I had students in my last class, no, I don't eat lunch.
No,
You don't eat lunch?
Late.
Well, so hopefully you had a little protein in your lunch. I had a salad with some chicken, so the chicken was the protein. My body is busy right now taking that chicken protein apart so we can use those parts to build up protein in my body that I might need for something, whatever protein I might need. And we need proteins for jillions of things, okay? But, you know, if you're trying to build muscle weight, right? If you're trying to add some muscle and building, you know, lifting weights or something like this, you're going to need protein to help build your protein, which is muscle. My protein I'm looking for are things like insulin and other things I need to make my body function properly. All right, there's kind of what a double helix might look like, right? We have the rungs of the ladder and the sides of the ladder right here. Okay, so what's the structure? There's a backbone made of sugar and phosphate groups. The sugar is called ribose. That's where ribonucleic acid comes from. You don't have to write this down. I'm just giving you some background information here. The stair steps themselves are called nucleotide bases, and there are 4 of them, and we'll see how those are put together. Information is stored in a series of nucleotide base sequences. It's a code. It's a code that codes for you, okay? And it's a code that codes for proteins. Only one side of the DNA is needed to figure out the other side. Okay, so our 4 nucleotide bases are, I'm going to write these down up here on the board. We have cytosine. We call it C. The board is wet, so you can't see it. We have guanine. This is not working. Guanine. Okay, thymine. And adenine. So we'll see my other one right here. So let's just remember them as a and a T and a C and a G. Okay? Cytosine, guanine, thymine, adenine. All right. And it turns out that A and T always connect together. So we have only 4 bases. Okay? Think about all the living things in the whole wide world. Think about the glory of humans, right? We are the apex of everything because we have the intelligence, right? And compare that to a mushroom. It's living, right? They all have the same type of DNA. In other words, just 4 different bases, but one of them codes for a mushroom, and one of them codes for a human. Crazy, right? But the DNA is all super similar. It's just what is the order of that DNA? And there's only 4 bases? That seems almost impossible, but they're long enough and big enough that we can have all these different combinations of those. But furthermore, A always attaches to T, it spells the word at, and C always attaches to G, that spells Christ is God, so CG. If you remember AT, at, then you'll remember the other 2, right? That kind of thing. And so therefore, if I have one side of it here, I can see what is paired up to it down here. So if I give you a thing like this with this code AGTCATCCGTAATGC, you can tell me what's the other side look like. So this one would be T, because A and T, what would this be? C, A, G, T, A, G, G, C, A, T, T, A, C, G. Okay, you probably have something on that quiz in a couple weeks to just show that the other side of it. Just remember A goes with T and C goes with G. Not that difficult.
I like it.
All right. There's what our double helix looks like. Okay, so here it is, deoxyribonucleic acid. Here's our bases, adenine, thymine always together, guanine, cytosine always together. And our backbone is a sugar called deoxyribose. Well, it's called ribose. Deoxy means 2 oxygens are off of it. Because there's another sugar called ribo. There's something called RNA, ribonucleic acid. It just has 2 extra oxygens on it. You've heard a lot about RNA lately, haven't you?
Right.
What have we heard about RNA? It's that virus. That coronavirus is an RNA virus. Yikes. Okay, so but we have DNA in our. It's the code of life, and we also have a phosphate link. So there's a sugar as our backbone and a phosphate link, and then these 4, 1, 2, 3, 4 nucleotide bases: adenine, thymine, guanine, and cytosine. You can remember those with A, T, C, and G. All right, it looks like this, however. Okay, it's very complicated, and it's organic chemistry. You can see all these little, aromatic rings. Every time you see a little corner right there, there's a carbon right there. They just leave it off so it doesn't take up so much space. So every angle has a carbon, unless there's something else, or like that N right there or that O right there. But there's a carbon there, carbon there, carbon there. So 1, 2, 3, 4 carbon ring. 1, 2, 3, 1, 2. That kind of thing. And this is what A looks like, and it's always going to link up to T. And our C looks like this. It's always going to link up to G. And it's going to make those rungs of that ladder. All right, let's watch a little video about it. See if it gives you a little help here.
DNA contains 2 strands of building blocks called nucleotides, arranged like a spiral staircase. Each nucleotide includes 3 parts: a phosphate group, a sugar molecule, and one of 4 bases: adenine, guanine, cytosine, or thymine. The sugar phosphate bonds form the double backbone of the molecule, the handrails of the staircase. But we find the genetic key to DNA in the steps of this stairway, the nitrogen-containing bases. These bases link up using hydrogen bonds in a very specific way. Adenine will bond only with thymine, A to T. Cytosine only bonds with guanine, C to G. While these basic pairings never change, the order of the pairs along each strand varies greatly from one species to the next. In this elegant design, we see how nature stores the instructions to build all living things.
All right, plus making those proteins. All right, here they are. Notice how they kind of they show kind of a shape right there. We kind of do that just to make sure we know they fit together, kind of like a puzzle. Y'all aren't gonna you're not gonna forget A and T, C and G, right? Okay, don't forget that. Here it is once again. I've got my sugar and my phosphate group as the kind of the base over there, and then the stair steps are the A and T and the C and G. Okay, how does an organism produce offspring that's very similar? And how does the small offspring grow into a large adult? So both of those things are commanded by DNA. Okay, first of all, the traits being passed to offspring, and secondly, growing, putting those proteins together every day. So we can see a bunch of living things right there. All right, genes. Number 1. I think you'll have a matching thing here, yeah? So number 1 is genes. Heredity and genetic factors are determined by genes, which is a basic unit of heredity. Our definition is a section of DNA that codes for the production of a protein or portion of a protein, thereby causing a trait. Do you see it on your deal?
Yes.
You're going to put a 1 by that definition.
Oh, okay.
You get it? Right. I kind of like that fact that that means you don't have to write as much, right? You've heard of genes, right? Someone will say, well, it's in your genes, you know. Your DNA codes for, you know, being a good basketball player or whatever you are, right? I don't know if that's exactly true, but I think talent does have some genetic aspects to it. You know, otherwise, you know, we'd all be professional ball players, right? You know, those football players out there, they have some natural talent, and then they develop that talent with practice, probably lots of hard work. Question, Riley?
No, I'm just trying to make sure you don't skip the...
All you're doing is putting a 1 in the blank that says a section of DNA that codes for the production of a protein.
Just like a basic unit of heredity.
Yeah, that's what I was thinking.
You're going to put a 1 there.
That's what I was thinking it was. It's just I was looking for the one that's next to gene.
Right here?
Yeah.
I think these are the words, right? A section of DNA?
I felt it was a hereditary.
Okay. I tried to write it down exactly like it is in the book so we wouldn't have any confusion. Okay, so let's take a look at a gene closer up. So here's my cell. Here's my nucleus. The DNA is in the nucleus. We knew that. Remember our big cookie we made? We know the DNA is in the nucleus. When it's time for the cell to divide, they'll turn into these things called chromosomes. Okay, so they kind of clump up and they divide in this kind of thing. That's what these look like, chromosomes right here. So if we take a chromosome and unwind it a little bit, there's a section of it that will code for something. We call that a gene. Okay, so that gene will code on that particular part of DNA, the ATCG will code for something in this particular case, some sort of protein. All right. It also may code for your eye color. Although I will say eye color is a little difficult because it's on two places on one gene and then a third gene. So that's why we have different shades of blue eyes, different shades of brown eyes, that kind of thing. So it's not as easy to guess as things like, you know, your thumb like this or your earlobes. Okay, a chromosome is condensed threads of chromatin in the nucleus of the cell. You're going to put a 2 by that one. Okay, this is when what happens in a chromosome. Typically, this takes place when the cell's about to divide. The DNA will clump up and get really fat. These threads, it's called chromatin. When it's sitting in the cell not dividing, it's called chromatin. And it looks a little bit like a bowl of spaghetti. I'll show it to you in a second. But when it's time to divide, it gets all clumpy. And then this one, when you see it in the X shape like this, that shows that it has duplicated. That DNA duplicates in order to duplicate the cell. Okay, if my cell's going to have a new cell, like for instance, our skin cells are growing all the time, therefore I have this process called mitosis taking place in our cells all the time. All right, so you can see our chromosome. We're going to unwind it again. We're going to see our DNA, right? There are these cool little things called histones that are helping to keep the fold in the DNA. The DNA is folded up, all right? And so our chromosome is when it gets very thickened like this. It turns from the chromatin to the chromosome, bringing us to chromatin. Look at that bowl of spaghetti right there. The long strands of DNA and proteins in the nucleus of the cell. This is when the cell is not dividing. Therefore, they're kind of in long strands like spaghetti, if you will. Okay, so that's number 3, chromatin. You got it?
Yes.
Long strands of DNA and proteins in the nucleus of the cell. This blue-purple bowl thing right here is the nucleus. One thing you need to know is the DNA never comes out of the nucleus, ever. The only way for it to get copied and sent out is if something else takes it out and takes it out. The thing that takes it out of the cell to get it copied into a protein is called RNA. That's the only thing that leaves the cell. Okay, leaves the nucleus. Notice those histones are keeping things folded up nicely in that DNA right there. All right, nucleosome is the octamers. I just want to show this a little closer to you. The histones right here, and they're called nucleosomes, and they're in groups of 8 apparently, called octamers. All right. All right. Some portion of the base pairs form genes. We know this already. Right here. Base pairs are going to form a gene. Those are those histones again. And now let's watch replication. The first thing it's going to have, my DNA here is going to unzip. It's unzipping, and then new base pairs are coming and matching in there like a puzzle. It's unzipping down here still. And new base pairs are going to come in. They're going to link up, and now I end up with 2 new strands of DNA. Where are those nucleotides coming from? From our food that we ate. If I eat meat, this is why it's important to eat meat, right? You got to get those nucleotides. You can get them elsewhere as well if you're a vegetarian. See, this is linking them together, this little machine here, this little red, this green machine right here. All right, so DNA replication is the process by which 2 identical DNA molecules are produced from the original. It unzips and forms 2 new molecules. That's number 4, DNA replication. Bless you. I have a little visual aid here that I'll use on the board up here in a few minutes. All right, we're going to practice a little transcription here. Did I want to do this now? Name the DNA nucleotides that match this sequence. Cytosine, guanine, thymine. What comes from those? But what matches up with cytosine? C goes with?
Guanine.
G, guanine. Guanine goes with cytosine. Thymine goes with? Adenine. This one, CAA, is going to end up being GTT, yeah? Guanine, thymine, thymine. I don't know why I stuck that one in there. Okay, here's our replication. I think I meant to take that out. Here's our replication. We have our original DNA. Look, it's being taken apart here. It's being unzipped, if you will. And then those new ones, those new nucleotide bases are coming in and finding where they fit. And then we're making our 2 new strands.
So the primase and helicase is unzipping it, and then the topoisomerase is putting it back up.
The polymerase right here is putting it. This one I think is holding on to it. And then these, the primase and helicase, those are enzymes. Guess what enzymes are? Proteins. So those had to be coded to by DNA too. So this all works together. You know, it's pretty fascinating stuff. All right, centromere, the region where 2 sister chromatids are joined tightly together. Okay, that's number 5. Centromere is the region where 2 sister chromatids. So remember I told you that the chromosome will look like an X shape once it's been duplicated. So we're looking at, so this is a duplicated chromosome. It's all bunched up, it's all fat, and now it's formed two. It's gone through that DNA replication, so it's got two now, and it's being held together by this little centromere right here. All right, and you can see right here during metaphase and anaphase in mitosis, it's going to use that little centromere there to pull those parts apart from each other so I can have cell duplication. That's what's happening. We're having it's reproduction, cell reproduction right there. Number 6, the chromatid itself. One of two identical copies of a replicated chromosome, so each side of them is called a chromatid, and where they're together, they're called sisters, twin sisters, joined by their centromere. All right, so that's just an easy way to get through some of the vocabulary in this module, okay, by doing that matching on there. You do need to know those things. So if I start asking you questions about the chromatid, you don't say, what's a chromatid? You know, so you know what I'm talking about, okay? All right, so here we go. Here's our nucleus of our cell. We have our DNA in there. Notice they're all X-shaped. That means they've all been replicated. And we're going to see, this is called a chromosome. This whole X-shaped thing is called a chromosome. And this, we're going to look at what is the chromatid. The chromatid is that. All right, and the other chromatid is that. That's the sister chromatid right there. We're just seeing right here, it's unraveling to show us that that's DNA in there. All right, that's just a way to show that. All right, so what is the process then of making a protein? This is the tough part today. All right, so let's watch a video to give us an idea.
If you could step inside one of your cells, you'd see something that looks a lot like a factory, building thousands of different molecular machines. It would have tiny assembly lines starting in the cell's nucleus and stretching out to structures called ribosomes.
We do that.
These assembly lines start with DNA and build RNAs and proteins. In a very real sense, you. DNA has all the blueprints for everything the cell will ever need to build. RNAs are assorted molecular machines that do all kinds of work in the cell. They carry components from place to place, ratchet up the speed of operations, switch other machines on and off, and relay coded instructions from the nucleus out to the factory floor. The other product, proteins, are strong molecular machines that do things like hold the cell together and send signals to other cells. There are two parts to each assembly line. The first builds RNAs and the second builds proteins. The process that builds RNAs is called transcription. It happens in the cell's central office, the nucleus, where DNA blueprints are stored. Let's say that a cell needs to make a transfer RNA, or tRNA, a triangle-shaped RNA which transports a building material called an amino acid. It starts at a tiny specific region of DNA with the code that makes the tRNA we need. This region is called a gene. A protein machine inside the nucleus pries apart the weak bonds that hold the two strands of DNA together. RNA building blocks swarm in and form a conga line complementary to the DNA. This RNA strand now needs to fold up. In the world of tiny cellular machines, shape determines function. RNA's 4 bases bond to each other and give the tRNA its 3D shape. And voila, our transfer RNA is ready for action. It floats out of the nucleus and picks up an amino acid. We'll come back to our tRNA in just a moment. Now let's look at the second half of the factory's assembly line, making proteins from RNA, a process called translation. Remember the scrape? This cell needs to make thrombin, a protein machine which helps blood clot and make a scab. Thrombin is just one of the tens of thousands of proteins your cells can make. Before translation begins, the cell transcribes a special type of RNA called mRNA, or messenger RNA. This mRNA carries the code for thrombin out of the nucleus and onto the cellular factory floor. Our mRNA drifts until it runs into a ribosome, a protein-making machine. The ribosome clamps down around the mRNA. tRNAs, just like the one we made earlier, drift in. The messenger RNA carries a coded message, which the tRNAs translate into amino acids, the language of proteins. This process, translation, proceeds down the mRNA, creating a chain of bonded amino acids. Just as with RNA, molecular shape determines molecular function, so these amino acids fold up into the protein's three-dimensional form. Finally, the ribosome comes to a bit of code that says, okay, we're done here, and releases its completed protein into the cell. So there you have it. DNA encodes genes to make RNA machines, and the ribosome translates special mRNA, which in turn makes protein machines. Unlike most factories, your cell uses just 2 processes to make tens of thousands of different molecular machines. It's like one factory that can build toasters, phones, cars, and even repair itself.
All right, that's simple, right? Well, not so much. All right, here we go. Think about it this way. If I have 2 things that need to be done, I have transcription and translation. So, transcription is the action or process of putting something in written or printed form. A court reporter's transcription of all the words spoken in court can be valuable during the appeal. Okay, do you understand that? It's transcribing it. I'm just writing down whatever you're saying. I'm taking dictation. Okay? Then translation is the process of converting those words or text from one language into another. Constantine's translation of Arabic texts into Latin. Well, that's an incomplete sentence, but yes.
Are we supposed to write
I've got
this down?
something easier for you right there. Okay. All right, so part 1 called transcription. The DNA unzips to reveal bases. So let me see real quick if this is what I want you to write. I think so. Sorry. DNA unzips. The messenger RNA. What you need to know in transcription, right? mRNA, meaning messenger RNA, acts like a camera to make a snapshot of the DNA. And it's going to take it to the ribosome. So what happens in transcription? The mRNA is going to read the DNA.
That's what we write down?
Let me make sure.
The part in the middle talking about how mRNA acts as a camera, that's already in here.
That's already in there?
Yeah.
Okay. Y'all, what are we on? 5A? 6A? Y'all just took 5. Okay, great. It already says something about the camera. RNA acts like a camera and makes a snapshot of the DNA to take the ribosomes to make proteins. It says that?
Yeah. RNA acts as a camera to the stem cells, the strand of DNA. mRNA, the nucleus, to go to the ribosome.
If yours was supposed to say that. So you might want to write down, make sure, see it says RNA, put a little M in front of it. Make sure you know it's messenger RNA right there. And then the negative image is made to be used by the ribosome. And there's something very important you need to understand, and go ahead and I guess write it right next to it here, or in that blank spot right there. Make sure you, in RNA there is no thymine. All right, if I'm looking at a nucleic acid, also RNA is not double-stranded like DNA is, it's single-stranded. So that's some major differences there. They have 2 more oxygens for one. And there's, instead of T, in RNA, the T becomes something called uracil or U. Uracil in RNA. Both T and messenger, transfer and messenger RNA. Okay, so do you understand transcription? I'm going to come in that the messenger RNA is going to copy a negative of one side of a DNA. Yes? One side. It's going to unzip, it'll do one side. There'll be something doing the other side too, you know, but we're just looking at the one side. So RNA just has the one strand there. Okay, so see how it comes in? Messenger RNA comes in. It gets those from the DNA and it's going to take it to the ribosome. No, actually we're in the cell here. This is the DNA. It's unwinding and it's going to come make that copy. And then I have my messenger RNA, which is going to then leave the nucleus. So transcription happens in the nucleus because that's where the DNA is. Okay? And now translation is going to happen outside the nucleus. Here they are. My DNA is unzipped. My messenger RNA is coming. Notice I have a U instead of a T there. Because RNA doesn't have thymine, it has uracil instead. So if I show you a strand of something and you see that there's A, T, C, and G in it, you say, oh, that's DNA. But if I show it says AU, C, and G, you'll say, oh, that's RNA. You'll be able to tell right away. Let's practice transcription now. So here I'm going to take my cytosine, guanine, thymine and transcribe it using a messenger RNA. So what is going to be? Cytosine is going to be? Cytosine is going to be guanine. Guanine is going to be cytosine. Thymine is going to be adenine. Now what about these though? Cytosine, adenine. Adenine is going to be guanine, what?
Thymine.
No, there's no T in.
Oh, uracil.
It's going to be, yes, guanine, uracil, uracil. Okay? You get it? So watch out for that trick. It'll be tricked there. All right, now translation part 2. The mRNA goes to the ribosome. You need to understand that. It leaves the nucleus and goes to the ribosome. mRNA goes to the ribosome. It carries on it something called a codon. It has the code on it. And it just turns out the codon ends up as a group of 3.
Yeah, I think that's
3 things.
Can we write that at the
What do you already have on there? Part 2, you have nothing? You have a bunch of blanks there?
Yeah.
Okay, first thing you're going to write down is mRNA goes to ribosome.
Well, there's like a mRNA and a tRNA.
Okay, so under protein synthesis part 2 translation, write mRNA goes to ribosome. In the ribosome, the tRNA comes in. Then write down there tRNA comes in to make something called an anticodon. It's going to go code against 3 of the base pairs, 3 of the bases, and make something called an anticodon that links to the codon. And so what I'm trying to tell you is 3 of these things add up to coding for an amino acid. Okay, it's in 3s. The codon and the anticodon are in 3s. Keep in mind that tRNA is just doing a different, it's the same molecule but doing a different function, and it's going to copy the mRNA. Do you get it? So if mRNA made a negative of the DNA, the tRNA is going to come in and make a negative of that, which would be a positive of the DNA, except it has U instead of T. Am I losing y'all? A long chain then of amino acids will be created, and a long chain of amino acids is called a protein. Remember, that's what we're trying to do here. We're trying to make a protein. This is called protein synthesis. And it is complex. So if it's taxing your brain a little bit, that's okay. It should. It's tough. This is one of the toughest things in the year, I think. I can't wait till the second semester when we just get to dissect things, right? We learn about creatures and dissect them. That'll be more accessible, I believe. But this stuff is super important here. Protein synthesis is very important. It's the stuff of life. All right, so here it comes in. Now notice I've got 3 bases pairing to 3 bases on my, this is my mRNA, here's my tRNA, and those 3 bases come in, they code, and particular 3s code for something in particular. In this case, uracil, 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 coded for something called lysine. That's an amino acid. Okay, there's about between 20 and 26 amino acids. And at least a dozen of them are essential. And what I mean by essential is you have to eat them. You can't get them, they're not made in your body. They're not stored in your body. You have to eat them, and the only way you know you're going to get all the amino acids is if you eat meat. If you choose not to eat meat, if you're a vegetarian, you just have to make sure what they call complete a protein. So make sure if you're going to eat beans, eat rice as well. That completes the protein. You get everything you need. If you're eating meat, you don't have to worry about thinking about all that stuff. You just eat the meat and you get it all. Red meat particularly. All right, so you see I've got a code here. UUU codes for lysine. CAC codes for valine. CUC for glucine or whatever GLU is, and serine and glycine and these. And it grows along chain. And the chain then has its own code, and the code is based on 3 of the bases. You get it? You see how my code now is coding for a new thing? And that long chain of amino acids becomes a protein that I need. I noticed one of the things on the video said collagen. You know, collagen's what makes your skin nice and supple, it makes your hair strong, this kind of thing. As we get older, our collagen breaks down and we don't make as much, and so that's why people like me get wrinkly, you know, and so I put collagen in my coffee. to hopefully reinvigorate my collagen production. Y'all are making plenty of collagen because you're young.
What if I consume collagen now?
What?
What if I can start to consume collagen
It'll
now?
make your hair really nice, and your fingernails will grow really strong. Yeah, it's good for you. But you're making it. Yeah, you're making it, and you're doing okay with it. Okay, now here's what you're going to write in those blanks on the bottom, y'all. All right, our mRNA carries the code to the ribosomes in the cytoplasm. My tRNA codes for the amino acid. Okay, here it is. mRNA carries genetic code to ribosomes. tRNA carries amino acids to its position, and therefore connects the amino acids together, making a protein. So the messenger takes the code to the new coding machine, which is tRNA transfer, and makes a protein. So what do I want you to remember about this? Remember that amino acids, a chain of amino acids, is a protein, okay? Oh, we gotta hurry.
Oh,
All right.
I'm not ready.
I am, by the way, recording this one because I say it's not here. So if you want it, I can send it to you. You just have to make sure and let me know, because I'll forget. Oh my goodness, we have a lot to do yet. You're not going to get out early today. Sorry.
Oh, we already getting done.
All right. Think in threes, like a triplet. The 3-base sequence of a DNA codes for an amino acid. The codon is a 3-base sequence of mRNA that codes for a specific amino acid. And the anticodon is a 3-base sequence of tRNA with amino acid attached to it. Kind of comes with it hanging on to it already. And then when those amino acids get close together, they bond together. The next question y'all have is, what nucleotide base replaces thymine in RNA? You tell me. Uracil, right here, uracil.
Oh yeah.
Are we done with this page? Okay, it's uracil. DNA is the set of something for creating proteins. Set of instructions. Those two blanks there, fill those in. All right, and I think the DNA sequences, y'all do those at home, okay? Y'all do those at home. Y'all are going to fill these in while I get stuff ready. Oops, there we go. That's that last thing for you. Y'all can do those complement strand of DNA, then our mRNA, then our tRNA, because we're going to do that in lab right now. So I don't want to take the time of class time to do that.
Can you go back to the one that says the anticodon and the codon one?
Can y'all put these numbers down first?
Yeah.
I don't know if everybody has.
Takes like 5 seconds.
This is good to study for review right there. Kai, you got those? Everybody good? Right. There?
Yeah, that one.
All right, and we're going to get started in the lab here. So y'all
this finished.